Hi, everybody. Today I'm going to be walking you through how to do our Ecology Near Me project, your 10-week semester-long project that will begin in week 1 and end in week 10 with a presentation. So I'm going to share my screen and walk you through what we will do in this project. If you haven't seen the video on the syllabus, you might want to watch that before you watch this, just so you can get a rundown of kind of like what things are supposed to look like in this class. But I hope that this clears up any confusion or misconceptions about the project. I'm going to be recording one of these for every project that we do, just so that you all have something to go back to get help on understanding like what to do. So here in our Biology of Marine Organisms Blackboard page, we're going to navigate. Again, we want to navigate over to our side menu, and we're going to talk about an assignment, the Ecology Near Me project. So we're going to go to the Assignments tab. And the very first one is this folder called Semester Project Ecology Near Me. So you can see that there's a very lengthy summary of exactly what you have to do in this project right here. You also notice that this is a clickable link. This is a folder, so you can click on it, and there's a link to our Flipgrid, and then that same instruction file is available as a doc if you need to see it in Microsoft Word or you don't want to have it online only. So I'm just going to go back to the page that has all the information on it and walk you through all of it, and then I'll show you some details of Flipgrid and things like that. So, semester project Ecology Near Me. This course is entirely online and remote, which makes it a little bit difficult for us to do interactive, field-based learning with you, which is kind of the whole point of this course. So Beth and I put our heads together and we came up with this idea that y'all are going to do a really cool project in your own backyard or near wherever you're living. Your own backyard is obviously not necessarily something that everybody has. So we're going to be sending every student a field ecology care package. And you've already got an email from Beth about that. And there's going to be a video explaining what all of the pieces of it are and how you can use them. You're going to use the contents of this package or other materials that you have available to you in your location to complete an 8-week field observation project focused on an ecosystem near your current home. So wherever you're living, you're going to go find an ecosystem and you're going to do some data collection and observation in that ecosystem using quadrats and transects, which means that you are going to be sampling the benthos, which is the underwater ground, or the actual above water ground in an ecosystem, right? So you want to think about an ecosystem where you can actually quantify different organisms, plants count, that would be present on the ground in your field site. Again, you don't have to do this in the water. You don't have to do this underwater. You don't have to do this in a tidal area. You can do this in a grassy field or wherever, or even a parking lot if that's the only place you have available to you. We want you to go out and find an ecosystem that you can collect some data in, and then we want you to collect data. So what you're going to do is, and we have all these detailed expectations available below, you're going to turn in a weekly field journal and a weekly video update for weeks 1 through 8. And you're going to collect and analyze data in R, which I'm going to teach you how to do. And you're going to present your findings to the class. So generally, what we want you to do is go find an ecosystem, look around at it, observe in week 1, come up with a hypothesis that you might want to try to test. And then starting in week 2, you're going to collect data on that using our transect method. And there's a video on how to do that already, and we're going to show you a little bit more about that later. But essentially, you lay out your transect tape, you take your quadrat, and you place it in 3 random locations, and you quantify every single thing that's in there each time. And that's all you have to do every week. So when you do that, you record your data in your field notebook. You're going to draw some diagrams. You're going to think a little bit and write some responses. And then you're going to record a quick video update and say, like, here's what I saw this week. And it's just about a minute long. So we're expecting this will take you, 1 to 3 hours a week on this project, ideally. So this is going to be a big part of your grade. We just want you to understand how it all works. So the learning objectives for this project, like I've already said, are to explore how ecosystems change with time and disturbance. So you're going to go out and you're going to look at the same thing every week. And what Beth has been telling me is that, she lives in a rural area and she walks around on these same trails on her property every day, and she sees them change over time with seasons and with disturbance and for various other reasons. So the more you see the same area, the more you can observe kind of how things change transiently across time and try to figure out why that happens. And that's kind of the whole point of ecology in general, is to go and observe and kind of report out what you see and think about why things are changing and what might be causing them. So we're also going to hopefully teach you some field sampling techniques and ask you to practice and apply them. and then you're going to learn how to develop a hypothesis and design an experiment to test the hypothesis. You're also going to use some of your data analysis skills that you're about to learn. And you'll be practicing different types of presentation skills, including your 1-minute video pitch, a 1-minute summary, and kind of a formal research presentation. And again, you're going to write a report on the result. You're going to report out on your results in a 12-minute talk at the end of the project in week 10. So assignment details. During the first 8 weeks of the semester, you'll be observing a site near your home and identifying what organisms are present and how that site is changing over time and seasons, and how the site responds to a disturbance. So you're going to use the tools included in your ecology care package, which include the notebook, a transect tape, a quadrat, a hand lens, and a few other things that are going to be really helpful for you. And I'm going to try to answer questions about this project on the first day of class because you do have an assignment that is due that week. So a detailed rubric and a timeline for this assignment is included below. It's also included in the Word document that has the same information in it inside the folder here. So as you think through the assignment and think about how to set up your experiment, we want you to use our field trips that you're going to be watching through the course of the first 8 weeks of the semester as guides to understand kind of some of the concepts that you might want to apply in your presentation. But first, we're sending you out there basically with almost no background information that we've given you and asking you just to go look for stuff. Find something that you think is interesting, tell us what it is, come up with a hypothesis. This hypothesis is totally allowed to change, and then start recording data, and we'll see what we get. So I have this broken down by week. For every single week, I tell you exactly what you need to do. So for week 1, like I said, you go out to a location. That's it. A great option for this is if you're in an area where you're inland and you don't have access to the intertidal or to the ocean or to a lake or a pond or whatever, it's going to be harder, first of all, to record things underwater. So maybe you want to go ahead and do this on land. A great option for this is to start your transect at the base of a tree and move outward, where you can see a natural gradient of light and temperature and things like that moving away from a tree. But you can use any ecosystem near you, any ecosystem at all. If you're having a hard time figuring out what an ecosystem might be, please get in touch with me and Beth, and we will help you. So you're going to record your starting place in your field notebook. If you want, you can make a little GPS dot so that you can always go back to the same point every time. If you don't know how to do that, I'm sure you'll figure it out. Google Maps is pretty awesome as far as dropping pins. So you can do that using your iPad or your phone or whatever. So that's all you have to do in week 1. Go find a spot, pick a place, think about how you might want to work with it, and then we'll start recording data in week 2. So make sure that even if you pick a spot in week 1 and then change it, that's okay. But make sure that starting in week 2, you're going back to the same place every time. Like I said, you can choose any ecosystem near you. We are going to be asking you to record the presence, absence of anything that moves and the number of anything that moves of mobile organisms. So is there a mobile organism present? If so, how many? And then we're also going to be asking you to approximate the % cover of the ground area that's covered by sessile organisms, for example, plants. So in week 1, your goal is to pick an ecosystem and try to develop a hypothesis related to that ecosystem. How might that ecosystem change? Something like that. Hypotheses are welcome to change over the course of the term. Below, I'm giving you some ideas to consider when you're thinking about your hypotheses. And then here's a couple of videos on transect methods that can help you decide where you should put your transects. And these are actually some videos that are recorded by us and some videos that are not. So this first one is a video that Beth and I recorded. And the second one is a video of a super nerdy, awesome educator from the UK teaching us how to record things with quadrats. So hopefully, if you're tired of watching us, you can watch him and get a kick out of what he's telling you. So that's pretty much all you have to do. There's some additional instructions about how to count transects as we go. So that's week 2. You lay out your transect tape. Oh, sorry. That's still week 1. Final note on week 1, there's a safety note here. Please choose an ecosystem that you can safely access. Whenever you're going to go, day, night, whatever, make sure that that ecosystem is safe and that your transit to and from that ecosystem is going to be safe for you as well. Please consider daytime versus nighttime hours and what may or may not change across time. In addition, biology lab policy at Bowdoin states that you must have a buddy when you do any kind of field work. So please adhere to this policy. It's going to help you to have a second set of hands. Go out with a buddy if at all possible. All right, starting in week 2, you're going to actually collect some data. So the way that you do that is you get your transect tape out, you lay it down, and you randomly sample 3 to 5 quadrats along that transect tape. And I already told you for sessile organisms, you record the percent cover of that organism. And for organisms that move, you will count the number of organisms that are present in each quadrat. We're also asking you to identify these organisms to at least the genus level. So if you're having a hard time figuring out who is in your ecosystem, you can use the iNaturalist app to help you. For iNaturalist, what you do is you take a photo of the thing with your device, and iNaturalist will allow you to, once you take a photo, you upload it, and then you click like, what is this? And it will try to identify it using what photos of what's been identified near you, and also kind of a machine learning algorithm. So you should be able to get a good idea of at least what the genus of your organism is. If you're struggling with that, please, again, get in touch with me. I'm happy to walk you through it. And I posted the same videos for how to use a transect right here. So when you're recording your data, we want you to record the distance along the quadrat, or along the transect where you place your quadrat, and then you can count all sorts of stuff. There's lots of information here about how to do that. Weeks 3 and 4, you're going to do the exact same thing as week 2. You go out, you make your observations, you record your data, then you take a video. Here's what I found. You upload the video. You turn in your field notebook. That's it. That's all very easy. In week 5, I'm going to ask you to create a disturbance in your ecosystem's experimental plot. So I want you to do something that's going to disturb approximately 20% of your ecosystem. And this is going to vary by ecosystem. If you happen to be in your own backyard, maybe you can take the weed whacker and mow the grass, or you can dig a hole, or come up with something, anything. Introduce some rocks, something like that. Just change the ecosystem in some way. If you're not in your own backyard, you probably don't want to do something quite that destructive. If you're at a local park, it's going to be really hard because they may mow the grass every week, in which case you're going to have a disturbance all the time. So if that's the case for you, we can talk about this. But come up with something that's going to create a disturbance. Create some shade, pull some grass, move some leaves around, add something, remove something. It's up to you. If you're struggling with figuring out how to do this, please talk to me. One thing I don't recommend you do is create a fire. Don't do anything that's going to be dangerous for you. Please be safe. We'll introduce the idea of disturbances in class, and we'll talk about what you might be able to do in class and brainstorm. So I've given you a number of examples here, including the ones I just spoke about. So in week 5, you're going to continue recording the measurements that you recorded before, but now you're also going to do an additional measurement of at least one quadrat that is within your disturbed area. So how does your disturbed area compare to your not disturbed area? What's changing, what's not? And then week 6, 7, and 8, you're going to continue recording just as you did in week 5. You'll record your data from your disturbed area and your non-disturbed area. You'll write your lab notebook, and you'll take your video. That's it. In week 9, you're done. You no longer have to go to your field site. You're welcome to keep going and observing if that's a thing that you're really into. But we're going to give you all of week 9 to use your newfound mastery of R to add your data to a spreadsheet, read it into R, test your hypothesis statistically, and generate some figures that show us what you did and what you observed. And then in week 10, during my office hours and during class, during block 9, we're going to be having presentations. So hopefully people will be able to join us synchronously for as many of those as possible, but we'll be organizing a schedule in the near future where we can have people present during week 10. All we're doing during week 10 is presenting these presentations. Okay, that's it pretty much for how the project works. So if you have any questions about that, please get in touch with me or Beth and we can walk you through it. Again, this is a collaborative project that the two of us kind of built together, so both of us are happy to answer any questions at any time. Technology that you need for this. You need Flipgrid because you have to do a project, or you have to turn in a Flipgrid video for weeks 1 through 8. So make sure you have Flipgrid. You also should have iNaturalist. It's optional. If you're really great at using field books, or you already know what organisms are in your ecosystem, great. I don't. I take photos and look at iNaturalist all the time. So totally get that. You can also just take a photo and put it on your computer and go to the iNaturalist website and do it there. You don't have to have an app. Okay, grading. Every week, you're going to turn in a field notebook for weeks 1 through 8. Each week, you're going to get 10 points. Every week, you're going to do a Flipgrid video. That Flipgrid video is going to be worth 5 points. So that's 80 points and 40 points. And then your final project presentation, which there's a nice big long rubric below about how to do that, is worth 100 points for a total of 220 points. If you do this project and you do at least do all of the things every week. If you do all of the things every week, you're going to get a C in the class just for doing this project. If you do nothing else, pretty much. You're going to have to like try a little bit on a couple other things. But like this project is going to be a huge portion of your grade. So if you do well on it, you're going to do great in the course. And it's mostly pretty simple stuff. So all you have to do is participate, and you're going to get a pretty solid grade. I'm not here to fail you. I'm not here to make you really stress about your grade. I'm here to teach you, and I want you to learn all sorts of stuff and find it interesting and engaging. And grades be damned, essentially, as far as I'm concerned. Okay, if you try, you will do well in this course. Rubric for the assessment of each component. You can read about how we're going to grade you. So for your weekly field notebook, we're going to grade you on 5 different things. You'll get 2 points for each of the things. 1 is recording the date, time, people working with you, the location and the conditions, and any unique features you see. 2, you need to give us a species list every week of what you're identifying. And then 3, I need you to give us at least 1 detailed observation of a species or the interaction between species. That'll be worth 2 points. Number 4, a drawing of one organism complete with its scientific name, size, scale, and label at least 3 important anatomical features and describe their use, purpose for that organism. That could be tough because we're not going to be talking about every organism that you're dealing with. We're not asking you to be perfect or to know everything. We're just asking you to give it a shot. This is a really useful skill. And some of you who are really artistic are going to love having this idea and this option. So we're making it available to you because students tend to produce really cool stuff when we give it to them. A reflection, at least a paragraph in length, maybe 2. You can write it after you get back from the field if you want. On the diversity that you observe, hypothesize what key processes might be controlling it. So this is really a way for you to think about what's happening every week. And you can use these reflections to make your presentation at the end and to read, essentially, to make your Flipgrid video. One thing that we're not grading you on is your actual data. Make sure you are recording your data in your quadrats every week. Who's there? How many of them are there? If they're moving, how many of them are there? If they're not moving, what's the percent cover for every organism in your quadrat? Make sure you're recording that data because that's what you're going to analyze for the later parts of the project. All right, so every week you write that up, you turn it in on the assignments tab in Blackboard. Every week, 1 through 8, you record a Flipgrid video. They're going to be about a minute long. It's really simple. I just want you to show me what you did and what changed, if anything, and then tell me why. So the point breakdown, you turn in a video, you already have a 1.5 points. The video is at least 45 seconds long, you get another 0.5 point. If you talk about your hypothesis and update us on whether you think it's correct, that's another point. You get a 0.5 point for using terms and principles learned in class, and another 0.5 point for video or verbal evidence of ecosystem change. If there's nothing changing, you can say nothing changed. That's still a 0.5 point. Basically, if you turn in a video and you say words, you're going to get 5 points every week. So that's pretty great. All right, the last part, the 100-point part, is your data analysis and presentation. And there's a nice long rubric here, and I'm not going to read the entire thing out, but you can see that you can get a 3, a 2, or a 1 in every single category here. And there's a bunch of different sections that you have to have: introduction, methods, results, conclusion, design, organization, clarity, and how you handled questions. I'm going to grade you on all that stuff. We'll see how you do. There's a weight associated with each one too. So you multiply here, the weight is 3. You multiply your 3 times 3, that's 9. Here the weight's only 2, so 3 times 2 is 6. That's the number of points you can get out of each section. You can see that the result section is the biggest portion. So that's going to be your R. I want you to do good stats, and I want you to make nice figures. If you're struggling with that, I am here to help you. I like helping people with R. I'm so happy to do it. Please come to me if you need help. You can also talk to the TAs, who are both going to be able to help you as well. All right. At the end here, I've got a nice schedule about what you have to do every week and what's due every week. Please note that your due dates are Friday at 11:59 p.m. So in week 1, this is basically before we have our very first class. You need to go scout a location and show us an ecosystem you're going to work in. And then make sure you do your field notebook and your Flipgrid video. Okay? Hopefully that's going to be good. That'll get you started. And you'll note that basically every week you turn in your field notebook and your Flipgrid video by Friday at midnight Eastern, and you're good to go. And then the presentations are going to be due on Friday at 11:59 p.m. Eastern on the 8th of November, which is week 10. All right. That's it for me. That's it for this project. Hopefully that was helpful. If you have any questions about this later, please get in touch with me. I'm so happy to help you. And keep in touch and keep your eyes open for the next video about the other projects.